platforms available on the market are very accurate, they are still prone to error. Even at accuracies of 99% and greater, a sequence generated may contain incorrect nucleotides. This means that if a machine's accuracy is 99%, one base pair is read incorrectly out of 100. Since NGS platforms generate high amounts of output, these errors can add up quickly. The way to circumvent NGS platform limitations is to sequence nucleotides multiple times. The number of times a nucleotide is sequenced is referred to as coverage or depth. Coverage per genome can be calculated by dividing the total output generated in a sequencing run by the total size of the sample sequence. For example, running a human genome, which is approximately 3 billion base pairs, will yield approximately 333 times coverage on the Illumina HiSeq 2500, which has a max output of 1,000 gigabase pairs. Consult ABM's NGS must be validated quantitatively and qualitatively to verify if there's a sufficient amount of good quality DNA. What is considered a good quantity of DNA depends on the library protocol's specifications. Having either more or less DNA results in less efficient sequencing reaction runs. This generates low quality data due to read problems from flow cell saturation or reduced coverage because of insufficient DNA. In terms of quality, a good quality library is one that has a diverse set of DNA fragments with minimal duplicate fragments. This is important because during PCR amplification, duplicates of fragments will be generated. The consequence of duplicate fragments is that the sequencing reaction will be biased towards these fragments. Rather than having a wide range for DNA sequencing, and it is still used today extensively for routine sequencing applications and to validate NGS data, it utilizes a high-fidelity DNA-dependent polymerase to generate a complementary copy to a single-stranded DNA template. In each reaction, a single primer, complementary to the template, initiates a DNA synthesis reaction from the 3' end. Deoxynucleotides, or simply nucleotides, are added one after the other in a template-dependent manner. Each reaction also contains a mixture of 4 dideoxynucleotides, one for each DNA base. These dideoxynucleotides resemble the DNA monomers enough to allow incorporation into the growing strand. However, they differ from natural deoxynucleotides in 2 ways. 1. They lack a 3' hydroxyl group, which is required for further DNA extension, resulting in chain termination once incorporated in the DNA molecule. And 2. Each dideoxynucleotide has a unique fluorescent dye attached to it, allowing for automatic detection of the DNA sequence.
Cluster generation, sequencing, and data analysis. There are a number of different ways to prepare samples. All preparation methods add adapters to the ends of the DNA fragments. Through reduced cycle amplification, additional motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos. Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn, composed of 2 types of oligos. 2 types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over, and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in 2 single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small...
This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determine the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the index 1 read primer is introduced and hybridized to the template. The read is generated similar to the first read. After completion of the index read, the read product is washed off and the 3' end of the template is deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as Index 1. Index 2 read product is washed off at the completion of this step. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized and the 3' ends blocked. The original forward strand is cleaved off and washed away.
DNA is coiled into a shape called the double helix. The double helix can be unwound into a ladder shape. The ladder is made up of paired chemical letters called bases. Altogether, our DNA contains about 6 billion such paired chemical bases. There are 4 bases in the DNA alphabet: A, T, C, and G. In DNA, base A joins only with base T, and base G joins only with base C. To read the sequence of bases in DNA, samples are inserted into a sequencing instrument, where high-frequency sound waves break the DNA into smaller pieces that are only about 600 bases long. Special tags are added to the ends of the fragmented DNA. These tagged strands of DNA can then attach to a glass slide. In a sequencer, each piece of DNA is copied hundreds of thousands of times, which create tags. This sequence of colors reveals the DNA sequence of each fragment. Powerful computers piece together these individual DNA fragments and reveal the sequence of your DNA. Then a team of medical experts uses specialized software to analyze and compare the DNA sequences to identify the handful of variants that may be important for your medical care. Discuss the results of the sequencing of your DNA with your doctor and a genetic counselor.
DNA is packaged up in 46 structures called chromosomes. Your complete set of DNA is called your genome. DNA is made out of 4 chemical bases called A, G, T, and C. These chemical bases, or letters, are the building blocks of DNA. The human genome is made up of about 3 billion pairs of these letters. When you stretch it out, it looks a little like this. The exact order of these letters is called your genome's sequence. Genes are specific important parts of this long length of letters. Humans have about 20,000 genes. Genes are important because they contain the instructions cells need to make proteins.
On the correct sequence, Cas9 can cut the DNA. Its 2 nuclease domains each make a nick, leading to a double-strand break. Although the cell will try to repair this break, the fixing process is error-prone and often inadvertently introduces mutations that disable the gene. This makes CRISPR a great tool for knocking out specific genes. But making double-strand breaks isn't all CRISPR can do. Some researchers are deactivating one or both of Cas9's cutting domains and fusing new enzymes onto the protein. Cas9 can then be used to transport those enzymes to a specific DNA sequences are found in the cell. This could be useful for things like visualizing the 3D architecture of the genome, or to paint an entire chromosome and follow its position in the nucleus. CRISPR has already changed the face of research, but these new ideas show that what's been achieved so far could just be the tip of the iceberg when it comes to CRISPR's potential.
Myself Suchismita Sao, and I am from Centurion University. So here, this is my first workshop in biodiscovery. So I have learned so much about the sequencing, RNA sequencing and the gene editing that will help in my future. And overall, the workshop was very cooperative. And thank you.
My name is Lopamudra Mahanti. I am from BDG Autonomous College, Bhubaneswar. This was my second workshop of biodiscovery group. It will be helping my future studies. And here guidance is very cooperative and friendly. Thank you.
Myself Swarnanjali Parida. I am from BDG Autonomous College, Bhubaneswar. It was my second workshop with BDG group, and here I became to know about NGS, which is the most advanced technology now, and about CRISPR, and it will be very helpful in our future and for a good career, and the environment is very good. Thank you.
Hi, I'm Arpita Patnaik from BJP Autonomous College, and this is my first workshop under BDG. And it was the topic was next generation sequencing, and it was new for me, and I have learned many new things about genome editing too. And it was you were cooperative and friendly, and thanks for that. So thank you.
Hi, my name is Bandita Maharana, and I am pursuing my master's in BJP Autonomous College. And I came here to do a workshop under BDG, and I came to know about it. Like I didn't know about this. Under this, I did a topic on NGS. It was really nice. Here I went first day we came to know about RNA-seq, and on second day we came to know about DNA-seq. So of course I came to know about genome editing too. So it was really nice, and thanks to sir, he really cooperated with us. I came to knew about many new things. So thank you sir.